Kia ora koutou, ko Douglas Walker, toko ingoa, and I'm here with the wonderful Amy Rowe of Wellington Girls College. Amy's going to run us through the last in the Level 2 Biology external webinars. Today's one is on gene expression. It's great to see a number of you lined up and ready to start. If you've got any questions as we go through, please pop them in chat and I'll either interrupt Amy or I will save those till the end to ask. We've also got the Study It forums that you can visit, and I'll put the link into chat for those. So if you've got specific questions about any of your externals, then you can go over to there and teachers from across the country will answer those for you. All right, thank you, Amy. Over to you.
Thank you. Kia ora koutou, Level 2 Bio students. Apologies for having to postpone yesterday's seminar. I was unwell, but I'm looking forward to talking to you about gene expression today. So I'll just share my screen. There we go, and we'll present it. So gene expression, this standard is my favourite one. It sort of links all of the molecular ideas that we study in cell biology with the genetic ideas that we do in genetic variation. So it kind of provides a bridge between the other two standards. And it's what I did my research in when I was at university. So that's why it is my favourite. So there is probably less specific material in the genetic, in the gene expression topic compared to the other two, but it is a little bit more complex and a little bit more detailed. Broadly speaking, we can divide the topic up into protein synthesis, where we look at proteins, amino acids, transcription and translation, and then how we fold up our amino acid chain into a protein. Then we've got mutations, so what they are and how they can affect how a gene is transcribed and how that goes on to create a protein that may be different from the one that was intended, and then metabolic pathways, so looking at how genes and enzymes interact within an organism and the effects that it may have on that organism's phenotype. But let's get started. The proteins, we know that they're biological molecules. They're made of amino acids linked together in a big long chain. They are the most vital molecule in biology. Organisms are basically made of proteins, and proteins have many roles in a cell or organism. So these may be structural roles, there may be antibodies, enzymes for catalyzing reactions, transport proteins, receptors, lots and lots of functions for proteins. The instructions for the cell making the proteins are coded for by genes. And we know that genes are present on our chromosomes. They are sections of DNA that code for a protein. The DNA sequence is used by that cell to create proteins in a process called protein synthesis, which is just synthesis means to make something. So protein synthesis is making proteins. And it has three key parts. So the first is transcription, then we have translation, and we finish off with protein folding. Not everyone would consider protein folding a part of protein synthesis, but it is vital to actually making a functional protein. All right. So we're going to talk about protein synthesis first, and I've just put this very wordy and boring slide on here. These are just some example questions from some previous exams, and there is a fairly strong theme running through all of them. So this one up here, we're told that DNA, mRNA, and tRNA are all involved in the formation of proteins, and discuss the significance of them in forming proteins. 2017, discuss the relationship between DNA, mRNA, and tRNA and protein synthesis. And down here, just in last year's exam, talking about transcription and translation. But our first bullet point is a description of template strand DNA, mRNA, tRNA, the ribosome, and the polypeptide chain. So when we're talking about protein synthesis, you'll most likely get a question like this, which is basically stepping through the details of the process of protein synthesis through transcription and translation, and being able to relate the functions of DNA to the mRNA to the tRNA as well. And that's what we're going to do on the next slide. All right, so we know that proteins are made of chains of amino acids, so they're linked together by peptide bonds. So when we have that big long string of amino acids, we call it a polypeptide. Now, the sequence for that polypeptide is determined by the base sequence on our DNA molecule, on our gene. So in our gene, it's the order of those bases in the DNA that determines the sequence of the amino acids in that polypeptide chain. The sequence of the amino acids in the polypeptide chain is vital to the shape and the functioning of the protein, because when that protein is folded up, it is the order of those amino acids and the chemical reactions or chemical interaction between those amino acids that will determine the shape of our protein. We know that a cell's DNA is stored in the nucleus, and there's some really good reasons for that. It protects the DNA from chemical degradation in the cytoplasm. So the cytoplasm has many, many enzymes, free radicals, and other molecules floating around in it that would easily damage and degrade our DNA. And our DNA is very, very precious to the cell. It contains all of the instructions for everything that the cell does. So we need to protect that very vital DNA code, and that's why it is stored in the nucleus. Okay, so up here on the top right-hand corner, this should look familiar to you, which is the central dogma of molecular biology, and that's that a gene or DNA is transcribed into mRNA or messenger RNA, and that messenger RNA is then translated in the cytoplasm into a protein. And they used to say one gene, one mRNA, and one protein. Molecular biologists will now tell you that that's not quite true. But the general idea is that we start with DNA, it is transcribed into mRNA, and then translated into a protein. So the purpose of transcription is to make a faithful copy of the gene's DNA sequence and then to export it out of the nucleus into the cytoplasm, which is where translation takes place. So translation takes place in the cytoplasm, transcription takes place in the nucleus. Now, the reason that this is important is that the DNA cannot leave the nucleus. The DNA is stored in the nucleus so that it is protected from damage and degradation, and also it's very, very large. So once it is, or because it is stored in the nucleus, it's unable to leave the nucleus. It's unable to fit through the nuclear pore to travel to the cytoplasm. However, the cytoplasm is where translation occurs. So we need to somehow get our DNA sequence from our nucleus out to our cytoplasm, and that's where messenger RNA comes into play. So transcription involves the creation of messenger RNA, which is single-stranded, and sends it out of the nucleus and into the cytoplasm. So a bit of a recap on RNA. It's similar to DNA in that it is a reasonably long biological messenger molecule, and it is made up of nucleotides. However, RNA is considerably simpler to DNA. So for one thing, it is single-stranded. So where DNA is double-stranded and wound up into a double helix shape, RNA is just single-stranded. The name RNA stands for ribonucleic acid, whereas DNA is deoxyribonucleic acid. That first letter refers to the type of sugar that's involved. So in DNA, we have deoxyribose sugars, whereas in RNA, we have a slightly different sugar, which is a ribose sugar. So simpler. For our purposes, though, what we do need to know is that DNA is made up of nucleotides adenine, thymine, guanine, and cytosine, whereas RNA doesn't have any thymine in it. It has adenine, guanine, and cytosine, and the fourth nucleotide is uracil. So it takes the place of thymine. So anytime if you had thymine in DNA, you make a copy of it in RNA, you're going to swap the thymine for uracil. So there are three types of RNA we talk about in the standard. There's this one. The first one is mRNA or messenger RNA. As we know, it carries a genetic message from the nucleus to the ribosome. We have rRNA, which is ribosomal RNA, and it makes up part of the ribosome, which is responsible for translating our mRNA sequence into a polypeptide chain. And then we have tRNA or transfer RNA, and that's also involved with the ribosome and translating our mRNA strand into a polypeptide chain. Okay, so let's talk about transcription. Transcription starts at the promoter region of our gene. So our genes have three basic regions. The first one is our promoter, and this provides a signal to our RNA polymerase that this transcription should start in this region. We have the coding region, which provides the instructions, the triplets for our amino acid sequence. And then we have a terminator zone as well, a terminator region, which tells our RNA polymerase to detach from the gene. So transcription starts when the enzyme RNA polymerase binds to the promoter region of a gene, and the DNA double helix will unzip. Once the RNA polymerase has bound to that promoter region, it will move through the coding region and finish its terminator. Now, when the DNA double helix unzips, it separates out into two strands. And it can get confusing which strand we're talking about and which strand is involved in protein synthesis. So one strand is called the coding strand. And you might think, well, that sounds pretty important. This has the DNA sequence on it that needs to be copied, okay? But the cell has no way of copying that DNA sequence directly. So what we need to do is we need to make a complementary strand to our template strand. So the template strand is complementary to the coding strand. And if we make a complementary copy of our template strand, that will be the same sequence as our DNA sequence. So it's the template strand where our RNA polymerase works along that strand from the promoter through the coding region to the terminator, the template strand that the RNA polymerase binds to. Okay. So here is a basic diagram. I've got a few of them, so if this one doesn't work for you, there will be another diagram of transcription coming right up. We can see from this diagram that the direction of transcription, the direction that our RNA polymerase is moving in, is from left to right in this case. So this will be the area of DNA that has already been transcribed. This bubble here is where transcription is occurring currently, and here is presumably the rest of the gene. RNA polymerase has caused our double-stranded DNA to separate. We have our coding strand up here, okay, which isn't really involved at all. What is happening is that we have these free RNA nucleotides floating around in the nucleus, and they're able to match up with these nucleotides on our template strand, the blue strand of the DNA. And what RNA polymerase does is it links all of those nucleotides together. So you can see that this newly made RNA is a perfect complement to the template strand of our DNA. One thing to note is that where we have adenine on our DNA, usually the complementary on DNA would be thymine. However, because we're making RNA instead of thymine, we have uracil instead. Okay, so anytime that you would use a thymine in as a complementary to the DNA, you're going to replace it with uracil for mRNA. So, in wordy detail, the process of transcription involves RNA polymerase binding to the promoter region of a gene. The DNA double helix will unzip, and the polymerase works along the template strand. Free RNA nucleotides bind to the template strand according to the complementary base pairing rules. Cytosine binds with guanine and adenine with uracil. RNA polymerase works along the template strand, linking together the free nucleotides to create the mRNA transcript, or the mRNA strand. When RNA polymerase reaches the terminator sequence, transcription is completed, so that RNA polymerase will then detach from the template strand. The two DNA strands will then bond back together or anneal back together into their double helix shape, and the mRNA strand will detach from the template and is then transported out through a nuclear pore into the cytoplasm. Once in the cytoplasm, the mRNA will move to and attach to a ribosome, which is an organelle made from protein and ribosomal RNA. It's found in the cytoplasm, and it is our site of translation. So this is where the sequence on our mRNA is translated into a polypeptide chain, or a big long chain of amino acids. So as you can see in this diagram down here on the right-hand side, and this one here, our ribosome has two subunits, okay, a small subunit and a large subunit, and our mRNA strand threads or fits between them like a ribbon. And this is so our ribosome can read the codons along the mRNA strand. So our ribosome is able to detect our mRNA strand in groups of 3 nucleotides. This is what we call a codon. Translation always begins with the first codon on all mRNA strands, which is AUG, known as the start codon. That codes for an amino acid called methionine, or MET. So our ribosome is able to locate that start codon and begin translation from that point forwards. Each group of 3 nucleotides, or each codon, codes for a particular amino acid, and the ribosome moves along the mRNA strand 1 codon at a time, and it has delivered the amino acids by a third type of RNA called transfer RNA. So here is our transfer RNA. Usually it's said to be a cloverleaf-shaped molecule, which is this one here. Sequence here is all tRNA. The important part is, or it has 2 important parts. One is at the top here. This is where an amino acid will bind, okay, and it will be a very specific amino acid depending on the sequence of this area down here. So this area of our tRNA is called the anticodon, and it will have 3 nucleotides down here, and depending on which 3 nucleotides are up there, it will allow a very specific amino acid to bind at the top here. So our anticodon is a complementary sequence to the codons found on the mRNA transcript. So the mRNA transcript is read in groups of 3 codons, and the anticodon is the complementary. So if our codon on the mRNA transcript was UCG, then on our anticodon, the corresponding or the complementary sequence would be AGC. Did I get that right? So these transfer RNAs are free floating in the cytoplasm with their amino acids bound to them. And during translation, the anticodon binds to the matching codon on the mRNA strand and transfers its amino acid to the polypeptide chain. So I've got an animation of that coming up here. Okay, so we see this big pink blob here, divided up into a small subunit and a large subunit. This is our mRNA strand feeding through our ribosome. You can see that these transfer RNAs have their anticodon bound to the bottom and their amino acid bound to the top. It is difficult to make out the names of the amino acids on those tRNAs. They are being recruited in, they are binding to the complementary sequence on the mRNA strand, and the ribosome is then detaching the amino acid from the transfer RNA and linking it together into our polypeptide chain here. Once the transfer RNA has transferred its amino acid to the ribosome, which has formed those peptide bonds as part of that amino acid chain, our EMPs or used transfer RNAs detach from the mRNA strand and leave back out into the cytoplasm. What will happen is those transfer RNAs will then be able to bind with another amino acid and the whole process can start again. So again, in words, our mRNA strand threads through the ribosome, which reads the strand 1 codon at a time. Transfer RNAs carrying amino acids are matched to the mRNA sequence in order, with the anticodons complementing the codons on the mRNA strand. The ribosome links the neighbouring amino acids together with a peptide bond to form a polypeptide chain. So this tRNA will come in, it will sit down and bind on this codon. The two amino acids, this one and this one, will be right next to each other. So our ribosome will create a peptide bond between them, detach the amino acid from this transfer RNA, which will leave. And then the ribosome will shift along to the next codon. The tRNA detaches, the ribosome moves along the mRNA to the next codon, and translation finishes when the ribosome reaches a stop codon. And there are 3 different stop codons. Here is another example, same sort of diagram. I only included one because it was a diagram from last year's exam paper. So the purpose of this question was to label the different parts of this diagram. So here we have our DNA strand. We know that one of them will be a coding strand and one of them will be a template strand. You can tell that our mRNA strand here is being formed off this red strand, so this one must be the DNA template strand. You can see our mRNA strand is pictured leaving the nuclear pore, so this one must be our mRNA. Here is our mRNA strand, and there is this big globular thing, which hopefully you recognise as the ribosome. It kind of has these 2 lobes or 2 subunits to it. The cloverleaf-shaped things, or the things that are shaped like a T, is how I remember, are our transfer RNAs or tRNA, and the thing being made by our ribosome must be the polypeptide chain. Okay, so translation. As I said, each codon on the mRNA codes for a particular amino acid. And we can predict the amino acid sequence of a polypeptide chain by looking at the order of the bases on the codon and using a codon table. Okay, so the way we use the codon table is if you were to look at a sequence which was, let's say, AAU. The first position is A, so we start here with A. We're going to be looking along this line here. Our second letter is also A, so we were looking for the sequence AAU. So we're looking on A, and then we come down from our second position here to A. Okay, and we have 1, 2, 3, 4 codons to choose from here, so we're looking for AAU. This is the name of our amino acid, ASN, which is off the top of my head, I can't remember what that one is. Aspartic acid. Another example, you may have the sequence which is CAC. So we start here with our first position C. We're going to be looking along this big line here. Our second letter is A, so we come down from A, and then we look across C here, find CAC. Okay, and this is our amino acid, HIS or histidine. So if we are given an mRNA strand, we can use our codon table to predict the amino acid sequence that will be formed from that mRNA strand. Here's an example. Down the bottom here, we have our DNA template strand. Okay, ATG. We have transcribed it into mRNA. Okay, so the complementary to A is usually T. However, we've got mRNA, so it must be U for uracil. Complementary to thymine is adenine, complementary to guanine is cytosine, and so on. Okay, that sequence is then broken up into groups of 3 for codons. So if it is a group of 3 on DNA, it is a triplet. If it is a group of 3 on mRNA, it is a codon. And if it is a group of 3 on a tRNA, it's an anticodon. But once we have our codons, we can use our codon table. So our first one starts UAC. We can read off our codon table U here, A here, and we scroll down until we find UAC, and we have tyramine. So that's that one there. Okay. So with our sequence of DNA, we can transcribe it into our mRNA according to the complementary base pairing rules. And then once we have our sequence of mRNA, we can translate it into our amino acid sequence using our codon table. So once our ribosome has reached a stop codon, it will stop translating the mRNA, and it will detach, and our polypeptide chain will break away from the ribosome. Okay, at this point, we just have what's known as the primary structure of our protein, which is just our polypeptide chain. The polypeptide chain isn't yet a protein. What needs to happen is it needs to be folded up into a very complex shape so that it's able to perform its function. And the shape of a protein is vital to its function and whether it will function properly. We start off when we talk about the primary structure of a protein. We have basically just a string of amino acids. Okay, it's often shown as like a string of pearls. What happens to that primary structure is then it is folded up or curled up, and those amino acids are linked together into specific shapes by hydrogen bonds. And the two main types or shapes that we talk about are an alpha helix, as we see here. It's like a spiral. Or a beta pleated sheet, which is like this one here. And if you were to take a piece of paper and fold it backwards and forwards a number of times, it would look like that. And that's the sort of shape that some areas of our amino acid sequence may take. A secondary structure is still not a protein. We'll usually have to go through a third type of folding, which gives us our tertiary structure. So this involves the protein folding into complex shapes, and this is due to attraction and repulsion between the different amino acids. And the tertiary structure is held together by disulfide bonds, so a different type of bond. We will see that there are alpha helices and beta pleated sheets as part of this tertiary structure, and they are held in place by those disulfide bonds. And for many simple proteins, this might be as far as we need to go. This, depending on what it is, could certainly be a functional protein that is able to catalyze a reaction if it's an enzyme, or be involved in transporting molecules across the cell membrane, for example. However, for other proteins, we have what's known as the quaternary structure. So the quaternary structure, or fourth structure, is not all proteins have a quaternary structure, but sometimes a protein might need to be made up of several different polypeptide chains, or several different proteins in our tertiary structure. So when it requires several different proteins, they are then threaded together and bound together into our quaternary structure. So this would be made up of two different tertiary structures in this case. And the example given here is that hemoglobin, which carries oxygen in our red blood cells, is made up from 4 different polypeptide chains, all bound together. So those 4 different polypeptide chains first will have started out as a chain. Okay, they would have then needed to have been folded into alpha helices and beta pleated sheets, and then gone through tertiary folding. And once those 4 different proteins or subproteins were folded up in a tertiary structure, they would have been bound together into their quaternary structure, or their fourth structure. And they would not have been as functional, or they would not have been able to perform their role if they were just individual tertiary folded proteins. Okay, so let's have a look at last year's exam question on transcription and translation. So transcription and translation are both needed to build a polypeptide chain. The question asks us to compare and contrast these two processes and justify why both are required to build a polypeptide chain. So we've got some bullet points there which can help us put our answer together. It wants us to start off with a description of a DNA template strand, mRNA, tRNA, a ribosome, and a polypeptide chain. And then we're going to explain what we mean by transcription, explain what we mean by translation. And those will just be quite detailed descriptions of the events that happen during transcription and translation. And then lastly, we're going to justify why both processes are required to build a polypeptide chain. Okay, so I apologise, the next few slides are really wordy. I think my slideshow has been linked on YouTube so that you can have a look, and you may want to look through it in your own time. But anytime we're going to be going through an exam question, it is going to be a bit wordy and a bit boring. Let's start off. Let's start by describing our DNA template strand. It's the DNA strand that the mRNA is made from, and it is complementary to the mRNA strand. The mRNA is our messenger RNA. It's a short single strand of RNA that's made from the template strand. Our transfer RNA is complementary to the mRNA. It has an anticodon that matches with the codons on mRNA and ensures that the correct amino acid is added to our growing polypeptide chain as the ribosome reads the mRNA. And the ribosome is the cell organelle that reads the mRNA, and with help from tRNA, adds amino acids to a growing polypeptide chain that corresponds to each codon on the mRNA until a stop codon is reached. The polypeptide chain is just the name we give for a string of amino acids joined together by peptide bonds and which will fold together to make a protein. So that first bullet point is basically just providing definitions for each of those 5 terms. All right, so next part, we need to provide an explanation of transcription. So this will just be describing the events and what happens during transcription from the start to the end, until the mRNA strand leaves the nuclear pore. The transcription is the process where mRNA is made from the template strand of DNA in the nucleus. This process starts with DNA unwinding into 2 single strands. RNA polymerase enzyme will then bind to the template strand of DNA at the promoter region. The enzyme then moves along the DNA, adding complementary RNA bases, according to the complementary base pairing rules, until it reaches a terminator region. The completed strand of mRNA will now leave the nucleus through the nuclear pore. The 2 strands of DNA will then anneal back together, and this is the end of transcription. So with practice, you'll be able to get really concise with your descriptions of what's happening during transcription and translation. But what I would caution you on is to try not to lose any of that detail. So it's the detail that we're looking for, or that markers are looking for when they're marking this question or this type of question. Okay, so now we're going to talk about translation. Again, we're just providing an explanation or a description of the events that happen during translation. So translation is where mRNA is read by the ribosome in the cytoplasm, and an amino acid chain is created based on the codons on the mRNA. The ribosome binds to the start of the mRNA strand at the start codon, which is AUG. The ribosome will move along the mRNA, reading it 3 bases at a time in what are called codons. Transfer RNA with 3 bases, called anticodons, that are complementary to the mRNA codons will then enter the ribosome, will match to the mRNA strand. The ribosome will then transfer the amino acid from the tRNA onto the growing polypeptide chain and attaching them together with a peptide bond. Transfer RNA will then leave, and another transfer RNA enters the ribosome, again transferring its amino acid. The amino acids are joined together by the ribosome to form a polypeptide chain, and this process will continue until the ribosome encounters a stop codon. The stop codon tells the ribosome that translation has finished, and at this point the mRNA will leave the ribosome, the polypeptide chain will also detach, and this is the end of translation. So again, you can practice your descriptions and being as concise as you can, but making sure you're still including a lot of the detail in there. Okay, so the last part says to justify why both processes are required to build a polypeptide chain. Okay, but instead of doing that, we're going to make sure we reread the question every time we go to answer one of those bullet points. And the first part of the question says to compare and contrast the 2 processes of translation and transcription and justify why both are required to build a polypeptide chain. So I think in last year's exam, lots of people went through to this 4th bullet point, justifying why both processes are required, but they forgot about the main point of the question, which was to compare and contrast translation and transcription. So it's a good tip when you're answering exam questions and you're using those bullet points after you have or after you think you have finished writing your answer and you've addressed all of the bullet points, return and reread the question and make sure there's nothing extra there that you needed to do for your answer before you move on to the next question. All right, So comparing and contrasting transcription and translation. Comparing is finding ways in which they are similar, and contrasting is finding the ways in which they are different. So transcription and translation are both required to build a polypeptide chain. They are similar because they use complementary base pairing rules, and those complementary base pairing rules are vital to how they work. So in transcription, it ensures, or the complementary base pairing rules ensure that the mRNA strand is a faithful transcript of the DNA sequence. It's a complementary sequence to the DNA. During translation, the complementary base pairing rules are used to match the anticodons to the mRNA codons. So the complementary base pairing rules are vital to both processes. Secondly, both processes need a template to read off. So the DNA template strand is used as a template in transcription to build the mRNA off. And in translation, the mRNA is our template for building an amino acid sequence. So they both involve a protein reading a template to construct a new molecule. So that's the compare part. The contrast part means find ways in which transcription and translation are different. So they are different because transcription takes place in the nucleus, whereas translation happens in the cytoplasm. Nice, obvious one. All right. Then our last bullet point was to justify why both processes are required to build a polypeptide chain. Okay, so remembering DNA can't leave the nucleus. Okay, it's a gigantic molecule. It's big. It's not able to pass through a nuclear pore, and also DNA needs to be protected. DNA contains all of the instructions for making every protein that the cell will need. And it needs to remain whole and uncorrupted. So for that to happen, it is stored in the nucleus away from those degrading molecules that may be floating around in the cytoplasm. Transcription is needed to carry the DNA's message via mRNA to the ribosome in the cytoplasm. Translation occurs in the cytoplasm because that's where the ribosomes and the amino acids that are needed are located. So we need to get the instructions or the message from the DNA out into the cytoplasm where the ribosomes and the amino acids can be used to put the protein together. And the way that happens is via mRNA. Without transcription, the template strand couldn't be made into mRNA, so the instructions for making the protein would never reach the ribosome. Without translation, even if transcription occurred, there would be no process to form a protein. There'd be no way in which the information in the mRNA could be translated into protein. And for this reason, both processes are vital to build a polypeptide chain. So that sums up and answers the full question. So I will say before we move on, when you're addressing this question, as I said before, or the style of question, make sure you are answering each of the bullet points. And before you move on to the next question, go back and reread your question from the beginning again and make sure you have done everything that the question has asked. This part of the question up here, the compare and contrast, could be easily missed if you only answered the bullet points. Okay, let's have a look at mutations. We know that a mutation is a permanent change to the base sequence of a gene, and there are several different types. So the types of mutations you've probably heard of include a change in chromosome number or aneuploidy. So this is where chromosomes may fail to separate properly during meiosis, and an organism may have an extra chromosome or it may have one less chromosome than it needs, for example. You can have a block mutation. This might be where a section of a chromosome has been duplicated or may have been lost. It may have been inverted or turned around, or it may have been broken off one chromosome and attached to a different chromosome, which is a type of mutation called a translocation. These sorts of block mutations, because they are whole chunks of chromosome, will affect a number of genes. So it could affect all of the genes on that piece of chromosome that has been lost or moved. And lastly, we have point mutations, which is the ones that we look at much more closely in the standard. So point mutations are mutations at one point, which is why they're called that. They affect the sequence of a base in a single gene, and this is where a base or possibly a few bases have been replaced or substituted, may have been lost or deleted, or have been inserted or added into the sequence. So there are three different types of point mutations. The first one is base substitution. I will say that with the examples that I have given here, I sort of wanted you to understand the concept rather than using actual DNA sequence, because obviously there are no Fs in our DNA sequence and no Rs either. So this is just a short phrase broken up into groups of three that illustrates my point. For base substitution, this is where one or more bases have been changed. So in our sequence, the fat cat ate the rat. In our mutated sequence, that first A has been substituted for a T. Okay, so this no longer makes sense. This is not a word. FTT is not a word. So it has caused a change to our sequence, which in this case means that our sequence does no longer make sense. We can still get the general idea, but it's not obvious. So these types of mutations, base substitutions, might have no effect on our amino acid that is coded for. And we will talk about redundancy of the genetic code shortly, or it may have a minor effect. Base insertions, this is where one or more bases have been added to our sequence. So again, our sequence is the fat cat ate the rat. If we had an extra A in there, okay, after the first one, you would see that it corrupts or damages, changes the entire sequence after that point mutation. So all we've done is we've added one adenine in here or one A in here, and because our sequence is broken up into triplets, okay, the rest of our sequence doesn't make sense anymore. Everything has sort of shuffled along. This is what we call a frame shift. And we'll talk about frame shifts in more detail shortly. So a base insertion may cause a frame shift, which will affect every triplet downstream of the mutation. And then we have base deletion, which again can have a severe effect like our frame shift, like our insertion. So this is where one or more bases have been lost from the sequence. In this case, excuse me, our A has been lost and everything has shuffled up to take its place. So again, from the point of our mutation downstream or onwards, it has affected how all of the letters downstream are read. Okay, this is a frame shift as well. Okay, so frame shift or a reading frame shift is a serious consequence of an insertion or deletion mutation. Because the DNA sequence is read in triplets, adding or removing bases from the DNA affects how the sequence is read. So I'm going to give you a demo in just a moment, but this could be our DNA sequence up the top here, okay, and this underneath is what we call our reading frame. Okay, so if I come out of there, our reading frame, I'm just going to shuffle it up, okay, we'll divide our DNA sequence up into our groups of three. So these are our triplets, okay. Our DNA sequence is divided up into triplets, and they fit nicely within our reading frame here. If we were to make a change, or if there was an insertion in our DNA sequence and there was an extra G in here, okay, what it does is it shuffles all the rest of our sequence along, okay, and it shuffles them along within our reading frame. So the DNA sequence shuffles, our reading frame stays the same, but now those groups of three within those little frames are different, okay, and you can see what effect that has had down here. I'm just going to present that again. Okay, so this is the change we've made. We've added the extra G. Okay, these are our original DNA triplets. Okay, and you can see that now our triplets have changed from our second triplet onwards. So instead of GGT, we now have GGG. Instead of GTC, we now have TGT, and so on. This affected our mRNA codons, so I have transcribed it into mRNA already. We know that this one is going to be our start codon and this one should be our stop codon. And you can see that from this codon onwards, they have all changed. So all of our codons have changed as a result of that reading frame shift. This has a serious consequence for our protein, for our amino acid sequence. Okay, our original strand or our original gene started with our start codon, methionine. We had proline, glycine, and valine, and then we reached the stop codon. In our mutated sequence, you can see we've still got our methionine, we've still got our proline, okay, but everything else after that is a different amino acid. We have threonine, we have, I can't remember what that one is, glycine, and then we have valine, okay, so they have all changed from the point of our mutation onwards, okay? And because the shape of our protein is intrinsically linked, it's super important to the function of our protein, and it is linked to the sequence of the amino acids that make up that protein. If we change the amino acids, then we will drastically change the shape of the protein, which will drastically change the function of the protein. So mutations that result in reading frame shifts can have very drastic consequences for the function of the protein and render them non-functioning. Every triplet codon and amino acid downstream of a reading frame mutation will be different. Okay, this will result in a very different protein from the one that was intended and is likely to be non-functioning, so it won't work at all. Certainly won't be doing the job that it was supposed to do. A reading frame shift can also result in a stop codon being introduced too early, so that the protein is too small or it's not transcribed at all. Or the stop codon could be missing from the end of the protein sequence or occur much later. So the polypeptide ends up being much, much longer than it should be. Basically, the instructions are in the wrong order. One thing to note about reading frame shifts is that they occur if you have an insertion or a deletion of one base or two bases or four bases or five bases, etc. But if you insert three bases or six or nine or any other multiple of three, all we're doing is we are introducing a whole triplet or codon. Okay, so in the example here, if we add in those three bases to our DNA sequence, all it does is it shifts the other triplets along. But see how their reading frame hasn't changed. All we've done is we have inserted one new triplet. This results in inserting a new codon and inserting a new amino acid. And this may still have serious consequences for our protein. It may not function as it should, or it may have reduced functionality, but it's certainly not as serious a consequence as a whole reading frame shift. You can see that we have isoleucine introduced here, but we still have the GLN and the VAL amino acids as part of our protein. We still have our stop codon where it should be. Okay, so a reading frame shift doesn't occur if there are 3, 6, 9, 12, 15, etc. bases added or deleted to our gene. Okay, so let's talk about redundancy and degeneracy. They're kind of two sides of the same coin. So you might have heard the phrase that the genetic code is degenerate and therefore redundant, okay, but you might not be too familiar with what those terms mean, or you might need a bit of refreshing. So the two terms that are tricky are degenerate, which means, degenerate and redundant, but degenerate means a code in which several codes have the same meaning. So, for example, different codons are coding for the same amino acid. And the term redundant means the genetic code is redundant because many amino acids can be specified for by more than one codon. So, like I said, they are kind of two sides of the same coin. So several code words code for the same meaning, or several codons code for the same amino acid, and redundant means many of the amino acids are specified by more than one codon. But what does this mean? So we know that there are 64 possible codons, and that is a function of having 4 nucleotides and assembling them into combinations of 3. There are 64 possible combinations that we can make. One of these is our start codon, AUG, and 3 of them are stop codons. That leaves 60 codons left. And those 60 codons need to code for 20 amino acids. So some amino acids are coded for by more than one codon. In fact, most amino acids are coded for by more than one codon. And if you have a look at your codon table, you'll be able to see that in many cases, or some amino acids are coded for by sort of 5 or 6 different codons. Why is that important? Let's have a look at this exam question from last year's exam. So this is a question about mutation. It's a mutation in a membrane protein gene, CFTR, which causes cystic fibrosis. The template DNA sequence for part of the normal and mutated protein is shown in the table, and the affected bases are shown in blue. So this is our normal gene sequence for the CFTR gene, and you can see that these 3 bases have been underlined. This is our mutated sequence. Okay, there are no underlined parts here. If you count up your groups of 3 or your triplets, you've got 1, 2, 3, 4, 5, and over here you have 1, 2, 3, 4. So what's happened is that these 3 amino acids, these 3 bases are missing from our DNA template strand. Okay, they have been deleted. We are talking about a deletion mutation. So the first thing that the table wants you to do is to transcribe the DNA template strand into mRNA, and then to translate the mRNA into your amino acid sequence. So I've just done those rather than show you the codon table again. We've talked about how to use it. But here's our mRNA strand for our normal gene sequence. And this is our amino acid sequence translated from that mRNA. So 2-isoleucine, phenylalanine, glycine, and valine. Okay, and then for our mutated sequence, you can see already there has been a change here. So in our second codon, instead of being AUC, we now have AUU. Okay, then we have GGU and GUU. Okay, which are those ones here? Okay, but something's missing from the middle. Okay, because 3 bases have been deleted, 3 is our magic number, rather than causing a reading frame shift, all that's happened is those have been deleted and everything has shifted up that way. Okay, so our amino acid sequence, isoleucine, glycine, and valine. Okay, so there is 1 amino acid that has been deleted. Then the question asks us to discuss the effect of this mutation on the amino acid sequence and the protein. You want to describe the mutation, give an explanation of why the triplets TAG and TAA code for the same amino acid, explain what is a frame shift and how it affects the amino acid sequence, and discuss how this mutation affects the amino acid sequence and the protein function. Okay, so let's start with that first bullet point. The description of the mutation shown in table 2, as we said before, it is a deletion mutation. 3 bases, the GAA, have been removed from the DNA sequence, and they were split between codons 2 and 3, or triplets 2 and 3. As the 3 bases have been deleted, this means that a frame shift has not occurred. Okay, the triplet TAG has changed to TAA. Okay, so here TAG, these ones have been deleted, so this one has shuffled up next to the T and the A. So instead of having TAG, we now have TAA. Okay, the 3 bases, the triplet TAG has been changed to TAA, but both of these triplets code for the same amino acid, which is isoleucine, or ILE. This is due to redundancy due to degeneracy of the genetic code, which means that different triplets and codons can code for the same amino acid. So in this case, TAG and TAA both code for isoleucine. This will mean that the amino acid isoleucine, or ILE, is still produced. So that takes care of bullet point number 1 and bullet point number 2. Okay, so our description of mutation was that it was a deletion, and we talked about redundancy and degeneracy of the genetic code. Next, we need to talk about a frame shift. and how it will affect the amino acid sequence. We've already identified that this particular mutation is not a frame shift. However, the question has asked us about frame shifts, so we need to talk about it anyway. Okay, so a frame shift occurs when the number of nucleotides added or deleted is not a multiple of 3. This causes all of the triplets downstream of the mutation to be read incorrectly. Frame shifts can be deadly and usually more serious than mutations that don't result in frame shifts, such as substitution mutations. A substitution mutation or a deletion or addition of 3 bases will only affect one triplet or codon, and therefore only one amino acid will be affected. If only one amino acid is changed, this will change the shape of the protein, but the change might only be small. The protein may still function. It may just have a different specificity, so it's not going to be as specific for its substrate as it should be. In comparison, a frame shift will result in all the amino acids after the mutation to be incorrect. This result will result in the final protein having a very, very different final shape. And because shape is so important to the functioning of a protein, this drastic change in shape will mean that the protein is completely non-functional. A frame shift may also change the position of the stop codon, meaning that translation of the polypeptide chain is stopped early, or it will come too late, meaning that the length of the polypeptide chain is far too long. And again, this would also be a very significant change in the protein's final structure. Okay, so last point, discussion of how this mutation affects the amino acid sequence and the final protein function. So we mentioned that we are not dealing with a frame shift here. So the effect on the amino acid sequence is reasonably minor. We have lost one amino acid, but there must still be an effect on the protein's function, because we were told at the start of the question that the particular mutation we were looking at causes cystic fibrosis. So even though there is only a small change to the amino acid sequence, it does still have a significant effect on the phenotype, causes cystic fibrosis. So a deletion mutation, 3 bases have been deleted from the sequence, a frame shift has not occurred. The new mutated DNA strand is missing the triplet and codon, which codes for the amino acid phenylalanine, or PHE. This missing amino acid will change the shape of the final protein. Since no frame shift has occurred, the stop codon will still be in the correct place, so the shape of the protein won't be drastically different. However, the missing amino acid means that the shape of the final membrane protein has changed, and this change is enough to cause it to not function correctly. And this lack of function, or this change in function, has resulted in cystic fibrosis. Okay, so that is our 4th bullet point. And we have, in talking about the change in function, bringing it back to cystic fibrosis, we have linked back to the context of the question. So if you remember, the start of the question was we were talking about a mutation that causes cystic fibrosis. So we needed to somewhere in our answer talk about how that mutation causes cystic fibrosis. So I think I've got some tips for answering the mutations questions. Firstly, address the bullet points. You will most likely need to identify and name the type of mutation described, and in most cases it will be some type of point mutation, whether that is an addition, a deletion, or a substitution. Possibly there may be a frame shift, or there may not. Once you've identified the type of mutation, you need to describe what is the impact of this mutation on the DNA sequence. So how has the mutation changed the DNA sequence? How it has changed the mRNA strand, or the codons that are produced, how that will affect the polypeptide and the sequence of amino acids in the protein that's being synthesized, and bring it back to the context of the question by talking about how that mutation will affect the whole organism. And then, as I said earlier, when you think you're finished, go back and reread the question and just double check that you have answered the whole question. So not just the bullet points, but checking out, making sure you read through the whole question and that you have addressed every part. It can be easy to miss some of the key parts of the questions if you only look at the bullet points. Okay, so almost our last topic is looking at metabolic pathways. So metabolic pathways are the result of our proteins and enzymes working together. They are a series of linked biochemical reactions which are controlled by enzymes. The pathway begins with a specific substrate, which is then passed through a series of steps. All of these steps controlled by enzymes, where the product of the first reaction is, the substrate of the first reaction is turned into a product, and that product becomes the substrate for the second reaction. And the product of that second reaction then becomes the substrate for the third reaction, and so on. Each reaction in the chain is catalyzed by an enzyme. Because each reaction is catalyzed by an enzyme, and enzymes are proteins, each of those enzymes is controlled by a gene as well. So usually in these metabolic pathways questions, you'll be asked to talk about possibly non-functioning enzymes, which will be a result of mutation genes as well. And you'd be asked, or you'd need to link together the effect on the gene, the effect on the enzyme produced by that gene, and the effect on the metabolic pathway. Mutations in a gene for an enzyme can cause the metabolic pathway to end before it should. If the mutation causes the enzyme to change shape or to have an altered structure, then it may lose function or have reduced function. The function of enzymes is to catalyze the biological reactions, which are the steps in the metabolic pathway. The product of one reaction becomes the substrate for the next. And if one enzyme doesn't function, it can disrupt the rest of the pathway. Okay, if gene 2 is not functioning, then it is not able to take B and turn it into C. Okay, if C is not being produced because gene 2 is mutated, enzyme 2 is not functioning, it's not able to catalyze this reaction, C is not produced, then there is no substrate for enzyme 3. Okay, so as well as C not being produced, D won't be produced either. Because this reaction here is not catalyzed, because enzyme 2 is non-functioning, C won't be produced, but B will, because enzyme 1 is still working. So enzyme 1 is still busily turning substrate A into B, okay, and B will just start to build up because enzyme 2 is not able to catalyze the reaction that turns B into C. Okay, so if one enzyme doesn't function, it can disrupt the whole pathway, and this disruption can cause a buildup of the substrate for the non-functioning enzyme, and in some cases, this can be toxic. So as I said, if enzyme 2 is non-functional because of a mutation in gene 2, C and D won't be produced, but B will or may build up to toxic levels. Okay, so when we're talking about metabolic pathways, generally the easiest way to get our head around it is to look at lots of different examples. So here is a simplified metabolic pathway that makes red pigment for eye colour in fruit flies. And this was an exam question from, I think it was 2016 maybe. So you can see that this metabolic pathway has two enzymes involved, PTP synthase and PDA synthase. And each of those enzymes was coded for by a gene. So we have two genes involved. We have our initial substrate, which is PTP, and PTP is catalyzed by PTP synthase to turn into 6-PTP. PTP is our product of this reaction, but it is the substrate of the next reaction. So 6-PTP is the substrate for PDA synthase, and PDA synthase takes 6-PTP and turns it into drosophilin, which is our red pigment. It gives our fruit flies their red eye colour. When biologists expose red-eyed fruit flies to mutagens, they found that different mutations produce the same phenotype of colourless eyes in their offspring. Okay, and then we have our questions. The first one starts, define a metabolic pathway. So we will come back to this diagram, but let's move on and define our metabolic pathway. It is a series of biochemical reactions that are catalyzed by enzymes where the product of one reaction is the substrate for the next. Each reaction is catalyzed by a different enzyme. You might also say that each enzyme is coded for by a different gene. So this type of definition just wants you to explain what you know or how you know what a metabolic pathway is, but you may also want to link it to the fact that there are enzymes and genes involved. Definitely you need to talk about the enzymes involved, but you may also want to link to genes as well. All right, so the next part of our question asks us to discuss how mutations can affect the presence, absence, or amount of specific metabolic products in the metabolic pathway for red eye colour in fruit flies. We're going to provide a definition of what we mean by mutagen and a mutation, and we're going to give an example of a mutagen, and we're going to give an explanation for how genes influence metabolic pathways. So let's start with that definition of mutation and mutagen. Mutations are permanent changes to the DNA sequence of a gene, which can be caused by mutagens. Now, mutagens are chemical or environmental factors that can cause damage to DNA, and you just need to know a few of them. Okay, the examples: UV light, X-rays, or tobacco smoke, or you may know some others as well. Metabolic pathways are controlled by enzymes, which are coded for by genes. If a mutation occurs in one of the genes in a metabolic pathway, it can result in a poorly functioning or non-functioning enzyme. If the enzyme is non-functioning, it will not be able to catalyze its reaction in the metabolic pathway. Okay, so if we go back and look at our drosophilin pathway here, we know that if there is a mutation to gene 1, then PTP synthase either won't be made or it won't be functional. It won't be able to work. If it's non-functioning or not made, then it's not going to be able to convert PTP into 6-PTP. Okay, so our metabolic pathway stops right here. Okay, none of this is produced. Okay, and we may see a buildup of GTP. Okay, but GTP results in colourless-eyed flies. So we were asked, or where the question tells us that our biologists have noted that different mutations produce the same phenotype of colourless eyes in their offspring. One of the mutations must be to gene 1. If there is a mutation to gene 1, then the metabolic pathway can't proceed because PTP synthase can't catalyse this reaction. So we get a buildup of GTP and our flies will have colourless eyes. Gene 1 is functional and works and is non-mutated, but gene 2 is mutated. Okay, so our mutation is actually in gene 2. Then the enzyme coded for by gene 2, PDA synthase, will also be non-functioning. Okay, so gene 2 is mutated, so PDA synthase will also be mutated. It won't work. And if PDA synthase can't work, then it is unable to catalyze this reaction. So 6-PTP to drosophilin won't happen. Our metabolic pathway will stop here. Gene 1 still works, still produces PTP synthase, and PTP synthase is still busily converting GTP to 6-PTP. Okay, but that's where our metabolic pathway stops. So we would see a buildup of 6-PTP. Drosophilin would not be produced. 6-PTP is colourless, and so our flies would have colourless eyes. So if you have a mutation in gene 1, you'll have colourless-eyed flies. If you have a mutation in gene 2, you'll also have colourless-eyed flies. If you had a mutation in both of these genes, then you would still have flies that had colourless eyes. The only way that you can have red-eyed flies is if both gene 1 and gene 2 are normal and functioning, so that gene 1 is able to create PTP synthase, which catalyzes the reaction from GTP to 6-PTP. Normal gene 2 is able to create PDA synthase, and PDA synthase is able to catalyze the reaction from 6-PTP to drosophilin. And drosophilin is our red pigment that gives our flies their red eye colour. So the only way our flies can have red eyes is if both genes are functional. All right, so I've summarised, well, I haven't summarised it, it is very wordy and I apologise, but those notes are here. So red eyes in fruit flies are produced by a metabolic pathway in which GTP is catalysed to 6-PTP by the enzyme PTP synthase. The 6-PTP becomes the substrate for another reaction catalysed by PDA synthase and produces drosophilin, which gives the eyes a red colour. PTP synthase is coded for by gene 1. PDA synthase is coded for by gene 2. If there's a mutation to gene 1, PTP synthase may have reduced function or may not be functional at all. So it would not be able to catalyse the reaction converting GTP to 6-PTP. This would mean that there would be a build-up of GTP. There would not be any 6-PTP to be the substrate for reaction 2. Reaction 2 would not proceed and the metabolic pathway would stop. The fly would have colourless eyes as no drossopterin pigment is produced. If there was a mutation to gene 2, PDA synthase enzyme may not be functional at all. This would mean that the enzyme would not catalyse the reaction converting 6-PTP to drossopterin. This would mean there would be a build-up of 6-PTP, drossopterin would not be produced, so the flies would have colourless eyes. In order for the fruit flies to have red eyes, both gene 1 and gene 2 need to be normal or functional, and they need to produce those two enzymes to catalyse the reactions. If there is a mutation in either gene, the metabolic pathway will not proceed, and the flies will have colourless eyes. Okay, so when you're answering metabolic pathways questions, I have a few tips for you. Again, you want to make sure you address the bullet points. You want to identify which gene, protein, substrate, and product are directly affected by the mutation. You may be told that a particular protein or enzyme is non-functioning, so then you can deduce where the enzyme might be, or you might be told, where the mutation might be, or you might already be told that the mutation is going to be in a particular gene, and then you need to work out how that would affect the enzyme and the substrates and products in the metabolic pathway. You want to think about the impact of that mutation on the enzyme. How would it have affected the enzyme? The products that are made before the non-functional enzyme, or the product that will become the substrate for that enzyme that's not working, what effect will the mutation have on that? Think about the substrates and the reactions that are after the non-functional enzyme. Will they go ahead, or will they be able to proceed or not? And also link it back to the context of the question. What is the phenotype going to look like? As we did with our flies, we talked about whether the flies would have colourless eyes or red eyes. So as well as talking about the gene mutation, the effect on the enzymes being functional or non-functional, the effect on the metabolic pathway, and where our substrates and products would change, what would happen to them. Also link that back to the phenotype of our organism. If it's relevant, you might need to describe the impact of the mutation in each of the genes involved. So usually these sorts of questions will ask you about all of the possible different mutations that could occur to lead to a particular phenotype, just as our question did. It asked us about multiple mutations, all giving the colourless eye phenotype. So make sure you address all of the possible places that a mutation could occur and the impact that it would have on your organism. Which genes need to be working to achieve the described phenotype. And lastly, a good tip for all exam questions, when you think you're finished, go back and reread the question and check, have you answered all of it? To finish off, we're going to look at environmental factors and how they can change an organism's phenotype without changing its genotype. We know that the phenotype is the physical expression of the alleles inherited, and the genotype is the combination of alleles inherited. So environmental factors can influence how those genes and the genotype are expressed and how they may be expressed to give the phenotype. Almost every cell in your body contains a full set of DNA of your genes. Okay, they can contain all of the genes that your body will require. However, every cell will only use the genes that it needs at the time that it needs them. So if you think about something like a digestive enzyme like pepsin, okay, every cell in your body contains the gene for pepsin, which is a digestive enzyme that's used in your stomach and your intestine. However, not every cell in your body will use the gene for pepsin. The cells in your eyeball have no use for pepsin, so they will not turn on the gene for pepsin. They will not go through protein synthesis to produce the pepsin enzyme. They have no need for it. Similarly, your body will only use the genes it needs when it needs them. So an example of this would be the enzyme relaxin, which is released by the female body during pregnancy to release to help relax all of the ligaments supporting the uterus. The body has no use for relaxin most of the time, so the gene for relaxin is usually turned off, and only when needed during pregnancy is that gene transcribed. So the environment can influence which genes are switched on and when they are switched on, and whether those genes are switched on or off will impact the phenotype of the organism. So in many cases, our genotype can set the potential for a trait. So the example here is human height. You have the potential is encoded in your DNA and your genes for the possible maximum height you could reach based on your parents' genes and the alleles that they have passed on to you. However, environmental factors such as poor nutrition or a lack of sleep can affect whether you're able to reach that potential. Okay, so it may be that you have been exposed to environmental factors which have meant that your growth has been stunted, and you won't ever reach the maximum possible height that your alleles suggest you could reach. So that is an example where environmental factors have affected the phenotype beyond what just the genotype tells you. So again, we'll have a look at an example. So here is our Himalayan rabbit. They have an allele of the tyrosinase gene. So tyrosinase is an enzyme that is involved in the production of melanin. And melanin is a pigment that in rabbits makes their fur black or dark brown. Himalayan rabbits have an allele or a version of the tyrosinase gene, which makes the enzyme tyrosinase sensitive to body temperature. Okay, it means that it is inactivated at normal body temperature. It won't be able to catalyze its reaction. Okay, what this means for the phenotype is our rabbit's extremities, its nose, its paws, and its ears and its tail will be much darker than the rest of the animal. Okay, so for the body of our rabbit here, okay, it's very, very warm. Okay, it's normal body temperature. So because the rabbit has an allele where the tyrosinase enzyme is sensitive to heat and inactivated by it, the tyrosinase enzyme is not able to work in any of these warm areas of the rabbit's body. Okay, it's not able to catalyze the reaction to form melanin. So melanin is not produced in the warm areas of our rabbit's body, okay, which has made the fur white in those areas. The extremities of our rabbit, the ears, nose, feet, and tail, they are further from the rabbit's body, and so they are slightly, a degree or two cooler than the rabbit's body temperature. So they are cooler, and just that change in temperature or that difference in temperature will mean that the tyrosinase enzyme carried by this rabbit is able to function in those areas. So it is slightly cooler in those areas, so tyrosinase is able to work. It's able to catalyze the reaction to create melanin. Because the tyrosinase in these areas is able to create melanin, that melanin pigment accumulates and gives our rabbit the black nose, black ears, and black paws. The rabbit's phenotype is therefore a result of its genotype, okay, the fact that it has a heat-sensitive tyrosinase allele. It has the genotype for heat-sensitive tyrosinase, and it is a result of the environment as well, so the temperature at each of these points. There was an experiment done with these Himalayan rabbits where early in development, the back of our rabbit was shaved, and the researchers put a cold pack or strapped a cold pack to the back of the rabbit. It doesn't sound particularly nice for the rabbit, I have to say. And they left that cold pack there for a length of time until the fur grew back, and they were able to show that in that area of the rabbit where they had strapped the cold pack, the fur had turned brown or had gone brown. This is because that part of the rabbit where the cold pack was slightly cooler, It meant that the tyrosinase enzyme present was able to work as it was intended to create melanin, and the melanin became deposited in the rabbit's fur on its back. So the particular Himalayan rabbit will have had black hairs at its extremities and a big black patch on its back, again showing that the environmental effect of temperature has affected the rabbit's phenotype and how its genes were expressed. Let's have a look at one last exam question. The expression of a gene is dependent on many factors. This question is about an experiment on twin lambs and the importance of a diet in producing healthy lambs. Each set of twin lambs had functioning genes that were able to produce enzymes for energy metabolism and red cell production. However, within the two, one lamb had a diet that lacked vitamin B12, and the other twin lamb had sufficient vitamin B12. So vitamin B12 is a vitamin that we take in through our food and is responsible, has a role in the pathways involved in red blood cell formation and energy metabolism. So the question asks us to describe the difference between genotype and phenotype and identify the environmental factor that is affecting our lamb's phenotype. So the reason that we're using lambs or twin lambs, I should say, is because they are expected to have the same or very, very similar DNA. They will have the same phenotype. Okay, so a genotype is a combination of alleles that the lamb has inherited, and the phenotype is the physical expression of the trait determined by the genotype. So for our lambs, our twins will have the same phenotype. They'll have the same combination of alleles. However, they have different phenotypes. You can see in our lamb that lacks vitamin B12 that they have low red blood cell count, they have weak wool, and they have poor and stunted growth, whereas in our second lamb, which is receiving enough vitamin B12 in their diet, they are normal and healthy. They have normal red blood cells, strong wool, and normal growth. So the environmental factor affecting our lamb's phenotype is whether they get vitamin B12 or not, so whether they have sufficient vitamin B12 in their diet. So we are asked, give an explanation of why the study was conducted on twin lambs, the way in which each lamb's genotype is expressed, and then to discuss the relationship between environment, gene expression, and the resulting phenotypes of twin 1 and twin 2. So let's start on the first point here. So the study was used twin lambs because they will have identical or similar DNA, and therefore they will have the same genotype. This means that any difference in phenotype will be due to any environmental factors that are different between the lambs, and in this case, the environmental factor is their diet. Because the lambs have the same genotype, if they were put under the same environmental conditions, then the lambs should have identical phenotypes too. The phenotype of the lambs is different, and it's a result of the interaction between the environment and their genotype. Each lamb had the same genetic potential, but their diet prevented it from being realised in the vitamin B12 deficient lamb. A lack of B12 didn't change the DNA or the genes, but it did change the phenotype of lamb A. In both of our lambs, our genotype is expressed by the production of enzymes. Okay, and we know this because the whole purpose of genes is to code for proteins and enzymes. In lamb 1, without vitamin B12, these enzymes are unable to function, so lamb 1's genetic potential can't be realised. Instead, it has a weak phenotype with low red blood cell count and poor wool quality. In contrast, lamb 2 has the same genotype but has a different phenotype. It produces the same enzymes and it gets vitamin B12 in their diet. This means those enzymes can function as they should. So lamb 2 is able to reach their genetic potential. They're healthy, they have a normal red blood cell count, and they have strong wool. So this experiment proves that an organism's phenotype is not only the result of its genes, but also its environment. And that is the end of the slides, I believe. So you don't often, from what I can gather from looking at a number of years of exam papers, there is not always a question on the effects of the environment. But again, some tips for answering it would be to first address the bullet points, make sure you're able to identify what the environmental factor is that is affecting the phenotype, identify how genotype is contributing, and how the phenotype has changed as a result of the environmental factor. Make sure you're addressing those bullet points and describing how those factors are linked together. So linking the genotype to the expression of any enzymes or proteins that may be involved and how that has affected the organism's phenotype. And lastly, make sure when you think you've finished answering the question that you go back and reread the start of the question and make sure you have addressed every part of the question.
Thank you very much.
That's all right. I hope that was all right, and I hope that is useful to our Level 2 students.
Very comprehensive, lovely animations again. We've got a few questions that I think I've answered, but I'll throw them out to you as well. Do we need to write about splicing?
I'm not sure if I'm supposed to say this. I have marked this paper before, and there was no requirement to talk about splicing. I know a lot of teachers do mention it, but it is not usually specifically examined. So if you do want to put it in your answers, you're certainly not ever going to be penalised for it, but you may not be rewarded for it either.
Very good. And the other two, I think, really relate to the same thing. Is it likely we need to know about protein folding in great detail? And will we be needing to talk about the tertiary structure in the exam?
Not in great detail, no. I suspect the types of questions you're going to get and the way that you would address them would be to do with changes to the DNA sequence of mutations leading to a change in the amino acid sequence, which would lead to a change in folding. So you do need to know about the fact that proteins will fold up and that the way that they fold is vital to their function, but it's unlikely, I would think, that you would need to specifically go into any great detail around tertiary and quaternary structures of proteins.
Awesome. Those are all the questions that we've got. Thank you once again for that very comprehensive run through, Amy. And I'm sure the students will find that useful to watch it back over in the run up to the final exam.
I hope so.
And I'm sure people will get use out of it next year as well. So thank you again. Have a great day and really appreciate all the work that you've put into these 3 presentations on Level 2 biology.
Thank you. And all the best for the exam tomorrow, guys.
There we go. Stream has ended.